Now we have to move on to the second speaker, Shima Safaiyan. So Shima is originally from Iran. She's a postdoc researcher in the Max Planck Institute of Immunobiology and Epigenetics in Freiburg, Germany. Her primary research interests lie in the areas of immunology and neuroscience. So one of her favorite scientists is Santiago Ramon y Cajal, who was first to report the fine anatomy of the nervous system and was remembered as the father of modern neuroscience. So Shima is now going to present her recent work published in Neuron, White Matter Aging Drives Microglial Diversity. The floor is yours.
Thank you, Jerry. Okay, now I will share. Is it okay now? Everybody sees it?
Yes.
Okay. Hi, everybody. Thank you for such a nice event. I really enjoyed it so far. Yes, as Jerry said, I'm today here to present my data that recently got published in Neuron, titled "And White Matter Aging Drives Microglial Diversity." This work was done in German Center for Neurodegenerative Diseases here in Germany, in Munich, in the group of Professor Michael Simons. Before I start the data, I would like to first introduce a bit of my previous findings, because that previous findings basically was a kind of an insight to the new data that now we have in Neuron. During my other work, since I was working in aging, basically, what we found out is that in the normal aging in the brain, the myelin, which is actually a lipidic membrane around axon, starts to get fragmented. And this fragmentation basically happens by tearing off the complete part of myelin sheets from the axon and gets released into the extracellular space, and sometimes it can also be taken up with some phagocytic cells. And we could show that these extracellular myelin fragments is the number is increasing with the time in the normal aging. Interestingly, we could see that these myelin fragments are mostly taken up or phagocytes by microglia as resident immune cells in the brain. Here is just a summary of my previous data that shows that microglia are actively taking myelin fragments, which are accumulating in the aging brain. And while they are basically taking up myelin, this overload of myelin in the lysosomal system basically leads to some inclusions in the cell that are known as lipofuscin, and also at the end also induce some age-related changes by, for example, expressing some pro-inflammatory factors like MHC class 2 in the microglia. Therefore, when we finish this, the next question, the most interesting question, was to know, okay, what would be the difference of microglial responses between white matter and gray matter in the brain? Because it seems that in the white matter, when we have a lot of myelin and microglia are exposed to the myelin, they are changing. For example, they get aged. But then that was a very interesting question to answer. Microglial responses might differ between white and gray matter in aging. For that, we took advantage of a single-cell RNA sequencing analysis. And what we did, we isolate or we dissect white matter and gray matter from old animals. And then we sort the microglia, and we use two approaches, like one SMART-Seq2 for the low number of the cells, and the other one to validate our data. We also did droplet-based RNA sequencing. And then after that, using the UMAP, we analyzed the transcriptomes of single cells to be able to basically distinguish the gray matter microglia from white matter microglia. And then when we sort or clusters these cells, we end up with 4 distinct populations. 2 of them were homeostatic 1 and 2. These 2 populations were found mainly in gray matter and also in the white matter. However, there were 2 other population, activated microglia and white matter associated microglia WAM, that were specific to the white matter. About the signature of the gene signature of this population, I can help you go through, is that if you look at the WAM or white matter associated microglia, they are mainly expressing genes which are related to the phagocytosis, antigen presentation, meaning that they are quite active. However, they are basically the genes which are related to the steady state or homeostasis of the cells of the brain is quite upregulated in this population. This is the same for the activated microglia, but what is highlighted in activated microglia is that they are basically upregulating a lot of ribosomal proteins, and they are basically active for translation. Homeostatic 1 and 2, as you can see, they are opposite to the other two populations because they are not super active like the other one. They down regulate all these activation markers. However, they are quite in the homeostatic state. Next, we focus on white matter associated microglia WAM, and we wanted to know if this population, the formation of this population is somehow age related. For that, what we did, we dissect white matter and gray matter or corpus callosum and frontal cortex from the old and young animal. And this time we just send it for did basically bulk sequencing, RNA sequencing. And when we compare the upregulated genes in the white matter old with gray matter old, and also with young white matter, we end up with 39 overlapped genes. And these genes basically, these genes are basically overlap with the WAM signatures. Some of these genes are like galectin-3 or which is important for activation, CST7, important for the antigen presentation, CLEC7A, ITGAX, and ApoE. Okay, that was then for us clear that with this data, we can say that the formation of WAM can be age dependent, but we wanted to confirm this with the other data set from people, because nowadays people are really doing a lot of analysis on microglia in the steady state, in age, and also in the young animal. We took two data sets from Hammond and Fregerio, and there we reanalyzed the data, and luckily we could find again our 4 populations. But what we saw here is that our population in terms of timing are different. For example, activated microglia are always almost there. However, WAM comes up only with time or with age, basically. Therefore, so far we can say that the formation of white matter associated microglia or WAM is age dependent. Later, we dig a bit more in the gene signature of white matter associated microglia, because this is very important for us due to the myelin degeneration in the white matter. And when we compare we saw that we compared the gene signature with the other data set, we saw that these white matter associated microglia are expressing distinct parts of other population of microglia, which are known as DAM. Just for people who doesn't know about DAM, DAM is disease-associated microglia population, which is discovered by Crenshaw mainly in the neurodegenerative diseases, and it was in the beginning in the Alzheimer model. And DAM basically appears in 2 stages, stage 1 and stage 2. This is why it's DAM 1 and DAM 2. And stage 1 is a process that is and doesn't need a TREM2, which is an activation marker protein for microglia, but stage 2 needs TREM2. Therefore, as you can see, since we saw a similarity between our population and DAM2, we wanted next to know that if our population is also like DAM2, TREM2 dependent. To solve this problem, again, we run single cell RNA sequencing, this time in age TREM2 knockout mice. And what we could see after detecting our population, we could see that in the TREM2 knockout mice, both activated and WAM population are not coming up. And this was showing that with that, we could say that the WAM formation is not only age dependent, but also TREM2 dependent. and also TREM2 dependent, yeah, it also activated microglia were also showing that they are TREM2 dependent. Next, we wanted to know, okay, where are the WAMs? Basically, where we can find them in the brain. Here we used we did immunohistochemistry and also RNA-scope, and there we used some antigens or markers for which are already established for DAM, disease-associated microglia. These are galectin-3, CLIC7A, AXL, and 8GAX. And then we look for the microglia which are positive for it, and we call them WAM positive. As you can see, in barely you find WAM positive cells which are labeled with these 4 markers in all these quantifications. But if you take a look at the white matter in all of these bars, you see that there is a kind of how to say uneven distribution of the cells. Why, for example? Because some cells are only expressing as a single microglia, but some of these WAM positive cells are in the clusters of microglia that are known as microglia nodules. Therefore, we can say that WAMs are localized in the corpus callosum mainly and in the microglia nodules. But what are the microglia nodules? They are already explained in the disease situation like multiple sclerosis. But also in normal aging in the mouse, if you look at the cortex, you see that cells are quite individual, singular. However, if you go to the box B and C in the corpus callosum, you see that often these cells make some clusters together, like here 4 cells or here almost 6 cells, and they form like microglia nodules, and the number of these nodules is increasing with age, and we found the WAM population in these nodules as well. But what this WAM is doing basically, why they are there and why this cluster is formed. We did a correlative EM and electron microscopy, and there you can see here, for example, we have a cluster of myelin, which we correlated to the electron microscopy, and you see that these cells are always in the vicinity of this blackish compartment, which are basically degenerated myelin. And then in immunohistochemistry, we could see that these clusters are basically with some internalized myelin debris, which are labeled with myelin basic protein or MBP. And the quantification showed that that half of the cells which are with internalized myelin are existing in the microglia nodules. Now we want to know, okay, if WAM is depending on TREM2, what will happen to WAM when we knock out TREM2? Are they still formed or not? I mean, naturally, we should say no, they are not formed, and our quantification also showed that there is no WAM positive microglia in the TREM2 knockout mice. Then we started to further analyze these mice, and we realized, for example, in the TREM2 knockout mice, often the cells are in the homeostatic state because they express P2R12 or TMM111 a bit higher than control aged animal, meaning that they are basically stuck in the homeostatic and they are homeostasis, and they are not active anymore like control. In these mice, we also couldn't detect any microglia nodules. As you can see here, with time, even nothing comes up. Therefore, next question would be, okay, if in the TREM2 knockout mice, there is no WAM, there is no nodules, then what is happening to myelin? Because we could show that WAM is important for myelin removal. Yeah. What is happening, you can see that myelin debris or myelin fragments get accumulated in these mice, and they cannot be taken away from the environment by TREM2 knockout mice. We have more of these extracellular fragments of myelin in the TREM2 knockout mice compared to the control in time. Then the next question would be, okay, in the TREM2 knockout mice, what is happening to microglia when they cannot respond to myelin by forming WAM? Are they also altered in these TREM2 knockout mice? Yes, they are altered in a way that they are basically accumulating some inclusions inside the cell. Here again, this is another correlative EM and light and electron microscopy, and you see these black structures that they are inside the one cell of TREM2 knockout mice, and these cells with the inclusions are increasing in TREM2 knockout mice compared to the control. Also, the cells basically accumulating myelin inside. We might say that these inclusions are also relevant to myelin accumulation, meaning that TREM2 knockout microglia can take up myelin, but they cannot basically digest it. This is why it gets accumulated. They are also morphologically different than control because they have these fragmented or irregular processes, and the number of these cells is increasing again in TREM2 knockout mice. Okay, so far we learned a lot about WAM, that how it happened in the physiology and why it's important. Now is the time to basically study the WAM population in the pathological condition. Okay, but which pathological condition? If you hopefully remember, we mentioned that WAM signature is quite similar to DAM signature, which is disease-associated microglia, and DAM was first discovered in the Alzheimer disease. Therefore, we wanted to know if we also see WAM population in the Alzheimer, and the main question was to know what is the time sequence of DAM and WAM formation in Alzheimer. For that, we did another single cell RNA analysis by using FAD mouse models, 6 months old, and there we could not only detect our population basically, but also we could naturally detect the activated population. And then we use also other mouse models using the data set of Gregorio, and there also we could detect our full population plus DAM1 and DAM2, which I already introduced you. And then we did some time analysis, and we realized that interestingly, already at 3 months of age of an AD mouse, the population of WAM is coming up, but not DAM. This means that WAM basically is in the beginning starting to fall. But now I want to know if they are DAM and WAM both are coexist in the AD, how they are different from each other. For that, we took advantage of the characteristics of WAM, which is removing of myelin. What we did, we took a look at the young animal, a 6 months old animal, and we look at the microglial nodules, which are formed in the cortex, but they are not, in the corpus callosum, but they are not associated with the amyloid plaques, yeah? And these cells were again naturally active because they are expressing CLEC7 and also galectin-3, but more interestingly, already at 2 months of age, we could see that these microglial nodules are taking up myelin, and they are having myelin inside, and this is increasing even with time. The other strategy for us to know how different our WAM from DAM is to look a bit more into gene differential expression. We wanted to look for a kind of a gene that it can be used or a protein that can be used to distinguish between WAM and DAM2. And that was the best one, was a marker called macrophage migration inhibitory factor, MIF. And we compared the expression level of this marker, this gene, in 2 populations with the expression level of CLEC7A, which is basically expressed in both populations evenly. We had an antibody against it, and you can see that in the cortex, when we have amyloid plaques and macroglia gets around it, they are MIF positive. However, in the corpus callosum, when you have macroglial nodules here as well, also in the quantification, there is no MIF. This means that WAM and DAM are really different. However, but we wanted to know also, since WAM and DAM are both age dependent somehow, we wanted to know if also MIF is also coming up during aging. The answer was no. Actually, if you compare to CLEC7, there is no MIF coming up with time. Therefore, it can tell us that WAM and DAM are really distinct population in the AD model. Next question, one of the last question was to know what would be the further signaling pathway of WAM and DAM formation in the aged brain compared to the AD mice. We know that, it was already known that DAM formation in AD animal is depending on the ApoE pathway. Yeah, we wanted to know, okay, what is happening formation of WAM in the AD. Then we took a look at Fregerio data, where he had a mouse model of APP-PS1 crossed with ApoE knockout mice. We had reduction of, and okay, this is the single cell data sequencing, and we detected our population. We had a reduction of WAM in red and also DAM. Yeah, but still, this was not our data. We wanted to confirm that this is kind of validated. Then we set up again another RNA sequencing analysis for aged ApoE knockout mice, and there we interestingly see there is no difference in the formation of WAM and also activated microglia, meaning that this population is not related to or depending to ApoE. This we also confirm in the immunohistochemistry, where we use our antibodies, and we saw that WAM positive cells are also in the aged ApoE knockout mice. Yes, then with this, I'm coming to the summary. First of all, we could find a novel microglia subtype, which are white matter specific microglia. Two of them are activated and white matter associated microglia. WAM and these activated microglia are both TREM2 dependent, but only WAM is age dependent. Also, WAM formation is ApoE independent, and WAM gene signature is partially shared with DAM gene signature. And interestingly, both WAM and DAM coexist in the Alzheimer model, and they can be distinguished by the differential gene expression and also by their function. And one of the most important message of this work is that in aging, the activation of microglia might be dominated by forming of WAM, but later it can modify, it can basically shift to the DAM in the Alzheimer disease. And the last and very important again for us, which we're coming from myelin field, is that WAM formation might be a protective response toward age-related myelin degeneration. And with it, I want to thank you and happy to take questions.
Thank you very much. I remember your 2016 Nature Neuroscience paper. Yeah, I read that one. It's a very interesting idea. Okay, so now, yeah, I like to make this seminar more interactive. So I want to encourage the participants to turn on your video and ask your question directly if you like. So Eduardo, yeah, please go ahead.
Yes, I've many questions, but I'll just ask one since we're running out of time. Have you measured the presence of extracellular myelin fragments as well as intracellular fragments or inclusions in the AD model?
Not really.
I mean, does it look like aging AD or is it different?
Yeah. I mean, this is very interesting. No, that was a next step to go ahead, and there is another. I think right now, exactly, we wanted to know that how myelin is really affected in the AD model, but not that I did it myself. No.
Thanks.
Okay, if you want to ask a question, feel free to type in the chat or turn on your video. So I'm wondering if you know the role of this white matter associated microglia in demyelinating diseases like VAE or multiple sclerosis.
Yeah, I can say that these are a new population, of course, but the fact that they are forming is already shown kind of descriptively that these microglial nodules are drastically formed in the multiple sclerosis. And more interestingly for me, who are interested in aging, they are also detected, this data I think they are not published, but they are also detected in the aging human brain. Therefore, definitely, I can say that WAM has a contribution in the pathology of multiple sclerosis. Other disease, I don't know. I cannot really say. But for example, in AD, what we can say is that, as I said also at the end of my talk, is that it can be kind of a continuous stage of microglia activation that WAM forms at some point, kind of in a prime situation, and then when there is some disease situation like AD, then it shifts to higher activation, and then become like DAM, for example.
Okay, thank you. I think we may have two questions. Gustavo, would you like to be the first one? You are muted.
Yeah, thank you very much. Very nice talk. I'm wondering whether you know any relationship between WAM formation and inflammation in the AD model and in aging also?
Sorry, can you repeat the question? In the beginning, I just had a
If
bad.
there is any relationship between inflammation and WAM formation in the AD model and also in aging?
Like any correlation you mean between like inflammation? Okay. Yeah, this is a very big question, I would say. We still don't know what is really happening with the inflammation related to formation of microglia nodules or WAM, because here the interesting point is that basically these nodules are formed mainly in the corpus callosum, where still especially in the early stage, the AD is not the tissue is not super active affected, because the cluster or amyloid plaques basically form mainly in the cortex or in the hippocampus, but not in the corpus callosum. That's a very difficult question to make such a in AD per se. In aging I can answer, but in AD I don't know what is really the role of this WAM population, especially in the corpus callosum. But that's a very interesting topic that one should start to discover what is happening there with these cells.
Thank you.
And one should also first start, I would suggest one should start first, like the first question that our friend asked, to check how myelin is affected, because maybe this is already relevant to start the inflammation later, I hope I answered your question.
No, yeah, it's a very interesting topic. Thank you.
Sure.
Okay, thank you. The next question is from Guy. Please go ahead.
Hi, that was really very interesting talk. I was just wondering the relationship between the nodules and multinucleate cells, since microglia can multinucleate, and TREM2 has been shown to be involved in multinucleation of osteoclasts. So I was wondering whether your nodules are multinucleate, and whether nodules actually identifies WAM, or there are some cells that are WAM that are not in nodules.
This I really don't know, that if they are, I don't know how even to study that if they are multinucleated. Maybe you can give me a bit of insight how, because this is something that we just saw in the brain and we just describe it, and they are basically involved in myelin removal. But more than that, no, we didn't really go through to see how they are formed and what is the mechanisms and what is the relevance here. But if you give me some insights, that would be great to also understand better.
Well, I'll just have a look in the literature. I mean, a variety of macrophages can form giant multinucleated cells in a variety of conditions, and in some cases that can be TREM2 dependent, but it's not straightforward to tell the difference between a nodule and a multinucleated.
Yes, exactly. I also think that, because for me is like they are kind of deformed cells that sometimes you have difficulties to see how many nuclei are together, but I think they are very much, the morphology is also very much affected. And now that you mentioned this, I can tell you that now my next interesting project is to study what is happening to these cells and why the cells have the tendency to make such clusters. And I can believe that many factors, or especially in the cytoskeleton, can be affected. One of them can be vimentin, because vimentin is one of the genes that coming up highly in this population, and we kind of neglected that. Although some people say vimentin is also important for activation of microglia, but that can also, maybe that has to do something with also multinucleation, because vimentin is also important for making this pre-nuclear space quite to shape the cells, and then but this is something very interesting, and I want to work on that.
One characteristic of a giant multinucleated cells is they can phagocytose larger structures, and that's hypothesized to be the reason that they form in the periphery.
Okay, this I didn't know that such characteristics. Yeah, interesting.
And we've shown that when microglia multinucleate, they can phagocytose. They're more phagocytic and can phagocytose larger things.
Yeah, maybe they, in this way, they're kind of helping each other to get myelin, because myelin is also quite a big structure. Yeah, I mean, you see that they are really coming off these granules, or very big, these fragments, or can be big for one cell. Yeah, that's very interesting. Thank you for.
Thank you.
Maybe with electron microscopy, you could look at, I mean, since you've done some, and see whether in aging you could see multinucleated cells.
Yeah.
One way of.
Okay, great. The last question is from Sonia Kim. She's wondering if you know the origin of why matter-associated microglia, and if that's different from disease-associated microglia.
The origin, you mean, like, okay, the origin. We cannot really say. It would have been interesting to do like some fate map analysis, but we didn't do it. And I cannot really say certainly, and I don't want to guess where they are coming from, because of course we also had some hypotheses, but we just left it open for now. But that's an open question. I'm sorry that I cannot answer.
Okay, I think we're running out of time, and thanks again for both speakers, Anand and Shima. It was a great presentation and great discussion. And I would bring your attention to our next Thursday seminar, which is focusing on glycobiology. And I would like to invite more people. I hope more people will join us to host seminars, to present their own work, and then try to make open science more available to everyone in the world. And thank you very much. Thank you. I hope to see you soon.
Thank you for organizing. That was interesting. Thank you,
everybody.
Thank you.
Thank you. Bye.
Bye-bye.